So now we are going with the biotechnology. Okay, so let me give you first example so that you can understand better. So here are the two examples, A1 and B. So this is the human cell and this is bacteria cell. Okay, like this is the bacteria, this is the human cell. And now there are the two processes of the biotechnology. One is direct, like we can directly add a gene and to make any to use the biotechnology, and another process is the indirect, but we will use the indirect one. This is the car which takes the electron, this is known as vector. We'll discuss it later. So now as we know, here's the gene which we need, this is the gene of interest. So now we know here's the insulin, we need insulin. We know a lot of people are having diabetes and these all, so insulin is required nowadays. So for that production there are a lot of bottles and these all. So how that is actually made or again distributed in the bottles. So now insulin will be secreted by pancreas, but pancreas can even not secrete that much, so the bottles can be filled with a lot of amount. So what will happen, we will use here biotechnology, and not only insulin will be present in pancreas, it's present in our whole body, but it is not expressing itself, it will just only express in a pancreas. So that's the reason it is only present in a pancreas. And if we talk about the here, insulin will be given, and if we add this gene of interest, this the human insulin, this the human gene, and this is the specific human gene of insulin. We will add this into the bacteria. So this means bacteria will then grow, bacteria will grow and it will make insulin. So this means after that we can fill the bottles easily. This is the use of biotechnology in this world. Now if we talk about the host family, so now if we talk about here host, here's the bacteria, this is the host bacteria, they are having gene. Here is the plasmid. So now what is this, plasmid is a double helix structure, we can say, it's a double structure, or even we can say it's a circular. So now this is the structure. So here as I told you, we'll be taking this insulin. So here we will add this the bacteria part, this the bacteria plasmid. So we will add here the gene which we required. So what will happen, this will given to this bacteria, and after that this will produce. So this the process of the plasmid here and the transfer. So this is, and if we talk about here restrictions piece. So now what is that, here's the gene. Okay, so now when it marks a lot of cuts in that is known as restriction sites. Okay, so now here the specific restriction sites is given, that is electrophoresis. So this the specific restriction piece. What was in the restriction process, it was there's a one gene and we want to mark it, we just need a specific gene, we just need this insulin gene, we just need the another body part gene, here we need another body part gene, the specific gene, so that it will mark the pieces, it will mark or cut them. So that is known as restriction piece. And now if we move to the restriction, who give the concept and this all, sorry, there's the restriction. So the concept of restriction was given in 1963, and as we know, if we are having nuclease, so nucleic acid would convert this nuclease, nucleic acid will make this nuclease. If we talk about the DNA, so what will happen, enzymes cut DNA, enzymes will cut this DNA and will make the DNAs. If we talk about the RNAs, so that is also our enzymes will cut cut RNA and they will make the RNA. So here we are having like types of DNAs. There are like two types of DNA. So first one is exonuclease, second one is endonuclease. So now what was happening in the exonuclease, like in this, here's the gene, okay, here's the gene. So now if you want to cut, like here what will happen, the gene, like the restriction would be cutting from the end, like here it will start from this end, like this it will cut until this it will cut, like how much it need it will cut, like it will start from this end, like start from this end. If we talk about the endonuclease, so here's the gene, and if we need an specific, like if we need just this part, so this will only mark this one and cut into pieces. Here it will be cut from the end, and here it would cut from center, like from wherever it want, especially from the centers, not from the end. So that is endonuclease, and here we will talk about the endonuclease. So now if you move to an example, so like here we are taking an example. So this is the like bacteria, and here so now like here's the main DNA of bacteria, its main DNA, and here's the virus, like virus is coming in this one, like this is the structure of virus. And what virus is doing, like virus is inserting its DNA in this, and in this we are having restriction, like here's the restriction endonuclease. As I told you, we'll study here endonuclease, like to cut the mid between or like from where they want. So here's the restriction endonuclease. Here's the restriction nucleus. Here's the virus, it is inserting its DNA in this like cell body. Here's this is DNA. So now what will happen here, so now like this restriction endonuclease, what it will do now, it will perform its function, like this will distinct its virus DNA and cut into pieces, like it will cut from here, like this restriction, as I told you, its work is to cut from it. So it will cut, like it will not go inside, it will not allow bacteria, like it will not allow virus to cut bacteria or destroy this bacteria. So here this will be cut into pieces, this means this will be killed. So like antigens will be made like against this. So here restriction would be like this DNA would be cut by the restriction endonuclease. Now like by cutting this, adding this all process is known as restriction modification system. So this all is known as like restriction modification system. Okay, so this was all.